adenoma is around 15 weeks, early carcinoma 17, 18 weeks, and late carcinoma will go 20 weeks and on. And you can see in staining of the tumor, you can see the dysplasia and the adenoma, early carcinoma, and it's very easy to distinguish between them. And another thing that is nice about this tumor, it's actually modeling quite nice if you look in biomarkers. These biomarkers are also found in many of the human breast cancers. So this is a good model to ask questions about tumors and tumor microenvironment. So the first thing I did when I came is ask, okay, I'm interested in immune cells, and we have this nice model that tumors are spontaneously developed. What's happening with the immune system in these mice? And we have these results, which seems to be one figure, but this thing is about a year work of trying to take tumors from the animals and just asking what kind of cells I can find within the tumors. And you can see here the different stages, and here there's another stage, late adenoma, because sometimes it's very hard to find exactly the right timing and decide, oh, this is adenoma, this is early carcinoma. And sometimes it's really hard, so we add another step here. And what you can see here, that again, we can see a lot of different immune cells. But the thing that automatically jumped to my head when I did this slide is that we have here beginning stages. We have majority of the cells here, the percentage of the cells found immune cells will be monocytic, will be macrophages and NK cells. They are more monocytic. And in the late stages, the lymphocytes, which are the T cells and the B cells and the NKT cells, they are coming and they are basically not taking over, but they're becoming a more dominant population. So we have basically two stages. And this is immediately jump in my head that said, okay, we have something here that basically caused this recruitment, the later recruitment. And because we are macrophage lab, automatically macrophages came as a potential targets, a potential cell population that have a lot of influence on the recruitment and on T cells function in the later stages. And one thing about macrophages is that they are very, very diverse. Macrophages are found in a lot of our tissues. For example, Cooper cells in our liver, they are macrophages. Langerhans cells in our skin are macrophages. Microglia in the brain are macrophages. So macrophages are everywhere. And they have a lot of different functions anywhere they stay. And one of the things you can see, if you just take macrophages in tumors, you can find a lot of different functions of macrophages within the same tumor. We have macrophages that are related to invasiveness. They help the tumor cells to invade other areas. We have one that are actually part of the vessels, of the vessels, of the blood vessels. They are associated with metastasis. A lot of macrophages, so our lab actually showed that if you take the cells that are crossed from the blood vessel to the lung as metastasis cells, they're always accompanied by macrophages. They always go together with macrophages. Macrophages protect them and help them pass through from the blood to the lung. They are related to angiogenesis. They create a lot of factors. They create new blood vessels. They are immunosuppressors. They can suppress immune response. But they're also immunoactivators. They can induce the activation of the immune response, and they actually have the potential to kill cells themselves. So they do a lot of stuff, and this is what makes them unique, and this is what makes them an interesting target in the immune microenvironment, because they relate to a lot of different functions. So to explain how macrophages can influence the activation of T cells, we're going back to this kind of slide, and now we're talking about the two different ways of presenting peptides in T cells. So until now, the first slide I showed you, we talked about how all the other cells in the body present peptides to T cells. But there are particular cells of the immune system, macrophages, dendritic cells, B cells, they're called antigen-presenting cells, APCs. Okay? And then they have a unique ways to present peptides. What they can do, they can take the peptides from the outside, not only from the inside. So they can engulf in a protein, dead cells, apoptic body, take it in. Again, they are positive peptides, and this can be presented on class 1. It's called post presentation, and it can be presented on class 2 MHC, which is called class presentation. And then it's the same process, but the difference is instead of CD8 T cells, which are the cytotoxic T cells that actually kill target cells, it's in this case they present into a helper cell. Helper cells, T cells that are helping the formation of a better CD8 T cell activation. They are producing cytokines and supporting the activation of CD8. So this is two functions that macrophages can do. But I always said that T cells can recognize MHC molecules, and the molecule that in T cells is called T cell receptor that can recognize MHC, and this is true. But it's not, when there's interaction between APC, antigen-presenting cells, and T cells, it is not only one interaction between the receptor and the MHC, the T cell receptor and the MHC. There's multiple interaction of proteins. It's called the immune synapse. There's a lot of proteins that are all connected together, and somehow the T cell can sum all the signal it gets and decide if it goes to energy, if it goes to proliferation and differentiation, or it can go to cell cycle arrest and or even cell death. It really depends on the core signals, okay, that the T cell gets. It can get signals that can stimulate it, co-stimulatory, or it can get signals that inhibit it, co-inhibitory. And again, APC can do both. So now we're going to stop for the introduction for a second, and I'm going to start showing you some results again on my latest project. For that, I have to introduce you another mouse model. This mouse model is based on that we took the PMT model that I showed you before, and basically introduced another construct that is containing two peptides, class 1 peptide and class 2 peptide, a construct called 2A and a GFP. The 2A is basically a sequence that when this is translated, this will be translated and also this, and it makes sure that if we see GFP, we know that these two are also expressed. And what you can see here is a memory gland, memory tumor actually, of a PMT mice that express this antigen, and it's called PMT antigen mouse. And you can see here that the memory tumors, memory cells are green, the epithelium of the memory is green, and that means that we have the GFP, and also we have the expression of our peptides. This expression is specific in the memory gland. This is the regular mouse, this is the PMT antigen mouse, and you can see that this is the memory gland, this is positive control, and you can see here this expression, this is basically this yellow stuff. And it's very specific to the memory gland. So now we have a mouse that develops tumors, and the tumor develops specific antigens that we induced in, and we have T cells that recognize specifically these peptides. So now we have the antigen presentation, potentially, and we're going to have the T cells that can get activated and kill the tumor, potentially. We have both sides of the equation. So the first experiment is basically checking, okay, what happened in the mouse, in the memory tumor. And we have our mouse, we let it grow and develop tumors, and then we inject cells from donor cells. These cells are, we call them here now TCR, TCR T cells. They all recognize class 1 peptide that this mouse is expressing. And then after 1 to 4 weeks, we're going to check our T cells and we ask what happened to the T cells. And the way to find out about the T cells is basically the easiest thing to check if they get activated or not is to see if they're proliferated. Because the first step they're going to do is proliferation. So if they get activated, they will proliferate. If there's no activation, they're not going to proliferate. To do so, we take our T cells before we inject them and we part them with a fluorescent dye. The idea behind this fluorescent dye is that if they're not proliferated, it's very strong. Every time the cell will proliferate, they will lose part of the fluorescence because both daughter cells will have the fluorescence. So this is no proliferating, this is one proliferation, this is second proliferation, third, fourth, and so on. And we can follow about 8 to 9 proliferation rounds. And when they got to the point that I cannot follow anymore, I will describe them as completely proliferated. So now this is what we are expecting to see. We have the bone marrow, and the bone marrow will produce monocytes that will go to the tumor. They will pick up antigen. They will go to the lymph node. The antigen presenting, in this case macrophages, because we're going to follow macrophages, will present or not present peptides to T cells. They will activate them, maybe, and then the T cells will go from the lymph node back to the tumor and do what T cells do, kill. Or again, maybe not, we don't know. And this is basically the scheme that we're going to follow all the time now. I'm going to show this scheme probably 6 times. Okay? So the first question is our macrophages. I showed you before we have macrophages in the tumor. The question, what kind of macrophages do you have? So in order to do so, we use a method called flow cytometry. I hope you all know it. Basically what we do is take the tumor cells, tumors, isolated them to a single cell population, and then stain them with fluorescent antibodies. These antibodies can specifically recognize specific molecules that we are looking to see if they're expressed or not expressed on our macrophages. And then we go to the flow cytometer, and it gives us analysis and tell us if we can see fluorescent or not. And this whole state is basically related to one cell. We do everything simultaneous. We can see up to 12 fluorescent dyes. So we define macrophages by these markers. These are macrophages that are usually defined for macrophages. They are expressed CD45, CD11B, F480. They are negative for Lysis-C, and they are negative for Lysis-G. If they are positive for Lysis-G, it's granulocytes. So, and then we see what they are expressing. They are expressing class 2. This is negative, and this 2 is positive. They are expressing class 1. CD86 is co-stimulatory molecule. They are expressing. PD1 is co-inhibitory molecule. They are expressing. This is class 2 specific antigen. This is the antigen that we actually introduced in the tumor cells. And we can see actually the MHC class 2 with this antigen on it, on the surface of those macrophages. And we're actually checking here, it's all these molecules are surface molecules. They are on the surface of the macrophages, ready to be presented to the T cells. And then we have another co-activatory, and we have another molecule related to inhibition. So what you can see here that the macrophages are inhibitory or activatory is not really clear. And now we ask what happened here. What happened in the lymph node? Can we see in the lymph node activation of T cells? Can the macrophages present the peptide to the T cells? And again, our T cells are, all of them are tomato red positive, so we can easily find them. So this is tomato red positive cells. And this is a bit different way to show you what I showed you before in the proliferation assay. But what you see here, these are original T cells, original CD8 T cells of the mouse. And these are the cells that we injected. They are tomato red positive. Okay? Here, here, and here. And what you can see that the PMT mice, the regular ones, with no antigen, there's no proliferation. If you take 10 days after injection of the T cells, you don't see any proliferation. If you go 28 days after proliferation, after injection, we can see nice proliferation. And you can see here, we cannot distinguish our CSE staining, our proliferation marker, between the one we actually stained, our cells, and the cells that were normally in these mice. These mice have their own immune system. So basically what this says, that after 28 days, we can see a proliferation of the T cells. But the problem is that all of you know, when you're sick with flu, usually T cells, the one who basically make you better. But you are basically feeling better after week 10 days. So this is very delayed. This is not normal. So something is wrong already here in the level of activating the T cells in the lymph node. So we go another step. What happened here? Here is if our T cells can go to the tumor. And the answer, again, is kind of yes. After 28 days, we can see some of them in the tumor, not a lot. And again, this is something which we didn't expect, because the system here is supposed to be normal. These are normal mice, and we have a very delayed immune response, and very weak immune response. So again, we do the same thing. I show you exactly the same figures. But this time, we took a mouse that has no macrophages. We have I'll show you later, but we have a mouse here. So we have a mouse that we can induce the killing of all macrophages. Okay? So this is after 10 days. So this is the regular mouse, antigen mouse after 10 days with macrophages. And when you take out the macrophages after 10 days, we see proliferation. So suddenly, if there's no macrophages, we have a normal, or pretty much normal at least in a level of the time schedule of the immune response, we have a normal response. As you can see here, there's not a lot of cells. And I was very disappointed when I see this until, again, we go back to see if these T cells can go to the tumor. I saw this. The reason I don't see them in the lymph node is because they're in the tumor in a very large number. So now we know if we take out macrophages, we suddenly have a better, at least immune response in the level of activation and migration of T cells. So the last question that I want to show you today is if the fact that those T cells get to the tumor, they are better killers of the tumor. Now, the problem in our model is that we are working with a very late stage model. And also, if you see the mouse with the breast tumor, it's basically somewhere inside. It's very hard to measure. And also because it's very late, we don't inject so many cells, we don't expect to see dramatic reduction of tumors. So it will be very hard to find out if they are working or not. So we need a little bit complex mouse model. So what we do here, we try to see if we can inject our own cells into the tumor, cells that we can actually follow, okay? If they are dying or staying alive within the tumor. Then we can say about if these T cells are more active or less active. And this is the mouse model. It's a bit complex, so bear with me a little bit more. So we have, we take our mouse that express our peptides in the mammary gland, the one where we have also developed tumors spontaneously. We irradiate them to kill their own immune system. And we give them a different immune system. Either for a mouse that we can control the population of macrophages. Basically, if we treat with doxycycline, we can kill the macrophages, or a wild type. We let it recover, and then we start killing the macrophages, with the one, basically treating with dox, all of them, and we're going to kill the macrophages, all the macrophages in this mouse. And then, after 4 weeks of treatment, we will inject our T cells, okay, our transgenic T cells that recognize our peptide, and then we wait, and then inject into the tumor two populations of cells. Cells that are sensitive to our T cells, and cells that are not sensitive to our T cells. Each population of cells will have a different fluorescent dye. As you can see here, this is one population, one fluorescent dye, and another population, second fluorescent dye. And the ratio between these and these will be 1 to 1. Now, if you kill the mouse after 2 days and take the cells and isolate these cells from the tumor, and we can check the ratio. If the ratio stayed 1 to 1, there was no killing. But if the sensitive cells now, the ratio changed, we have less sensitive cells and more non-sensitive cells, means that our T cells would specifically kill the sensitive cells. It's a very complex to do, and it takes a long time, but then we get this. And what I can show you here is basically that if you take just the regular purity mice with no antigen, usually stays around 1. The ratio is fixed. The same is true if we have the one with the antigen with macrophages. When we take out the macrophages, we basically have 2 separate groups, a responder group and non-responder group. We have a non-responder group. These mice, simply, we didn't see anything. But we do have a group of mice that we can see an effect. And this effect is significant. We can see specific killing of our target cells. Now, there's a lot of reasons why we don't have this, all of them in responder. Mainly technical issues. This thing has to inject about 3 million cells. They have to go to all the system, get to the tumor, meet in a big tumor of 1 centimeter, about 50,000 cells that we are actually injecting, and then kill them. So this is very encouraging result. And what this led us to believe now is in this kind of model. That basically we have here acute reaction versus chronic reaction. So when we do have macrophages, they don't prevent completely the immune response, but they make it slower. They make it chronic. So non-completely fast, strong activation. We can see low activation after a long period of time. The immune system is there, but it's not very efficient. However, macrophages somehow, direct or indirect, they manage, when we take them out of the system, okay, we have much more acute response. And macrophages can do it in many ways. And we are now working, and we have interesting data now about trying to explain how macrophages do that. They can do it directly by influencing CD4 T cells or CD8 T cells, and they can do it directly by inhibiting the dendritic cells. And we think that when we take it out of the system, the dendritic cells now has the potential to actually activate better the T cells, and we can see a better and efficient immune response towards tumors. So, just a take-home message here is basically I showed you separate different things. One of them